I am very pleased to introduce today's speakers, Andrea Burfeid and Michael Kloster from the University of Duisburg, Essen in Germany. They are researchers in the phycology lab of Bank Pasteri, and they're a group leading some really exciting efforts in advancing machine learning for algal identification, as well as education. I learned about today's topic from a really nice blog post written by Andrea for the Young ISDR group. It's called Alternative to Microscopy in the Time of Coronavirus. So it's a very timely topic and I'm looking forward to learning more about it. So thank you both, Andrea and Michael, for joining us today. And Michael, please go ahead and start the presentation.
Okay, so hello everybody and welcome. There's a lot of different stuff going on in our lab, but the thing I can tell you about is how we are trying to boost diatom research by implementing and developing digital methods. And these are addressing stuff like standardizing optical microscopy and image quality, improving analysis, collaboration and teaching, enhancing morphometry, and upscaling abundance counts. I will detail all of this in the next 30 minutes, but for those of you who are in a hurry and need to leave early, here's a very short version. So here you see a guy who is doing his datum investigation pretty much like it has been done now for much more than a century without much change in it. So he's working bent over his microscope, straining his eyes, staring at the slide, identifying diatom cells and counting them. And now we want to take this very handsome man and transform him into a person like this one here, who is even more attractive. And I can claim so because actually both of them is me. And in this image, at least I'm having something like a decent haircut. And this guy here now is doing his diatom research using digital methods. And so he is sitting at his desk with the back very straight because his doctor told him to do so. And he is straining his eyes, staring at the monitors, watching all of the software he is using for improving and in parts automating his work so that he has more time for writing papers, writing grant proposals, and developing even more of the software which enables him to do all of this. Starting point for whatever we are doing is slide scanning microscopy, for which we are using commercial systems like this ones here. The reason for using such very expensive machines is that we want to standardize the imaging process and the image quality as much as possible, because that makes all of the downstream processing much, much easier, especially if you want to automate it. What we're aiming for are images like this one's here, so really high resolution, high quality, obtained by bright field microscopy using high magnification, high NA objectives. One technique we are always using when imaging is focus stacking, which just means that we are imaging everything in a series of slightly different focal planes, and then we will combine all of those into one single image where everything is in focus. And this now, once again, is much easier to process in the downstream. Through the years, we implemented several different imaging strategies, and we started with a very naive approach where we just scanned the slide in the form of thousands and thousands of slightly overlapping high-resolution images. This is very easy to implement, but also is very ineffective because, as you can see here or here, very often valves are somehow cropped by the camera's field of view. To overcome this problem, we then implemented a procedure which had been proposed already 15 years earlier by the ADIAC project. ADIAC is the so far largest project regarding automatic datum identification and classification, hence the name ADIAC, it's just an acronym of this. And they investigated all the tiny building blocks one would need to implement to have an end-to-end diatom identification pipeline. And one thing they proposed was the scanning procedure, where there was the problem that at their time they could just propose it, but they were not able to implement it because their self-built slide scanner did not have the mechanical precision required to do this kind of stuff. Nowadays, commercial systems have that, and so we were able to implement this procedure, which involves 2 scanning cycles, where the first is imaging everything just in a low resolution for the sole purpose of finding the location of individual diatom cells. And then exactly these locations are revisited in a second scanning cycle and then are imaged using a high resolution, high magnification objective. And as a result, you will then receive hundreds or thousands of images like these ones here, where usually there's a diatom cell directly in the center of the image. This worked really well, and we used this procedure for several years and also had a paper out explaining it in detail. But nevertheless, something like maybe 2 years ago, we then switched over to a third strategy, which is that we are now scanning larger contiguous areas of the slide in the form of a single high resolution image. So actually the scanning itself is exactly the same as with our very first approach, but now we are combining all of these individual images into one really big image which contains just everything. And this large image now is starting point for something which we refer to as virtual slides. And these are then extending the image data, at least optionally, by metadata describing the sample, plus by so-called annotations. And these annotations are describing individual objects, in our case diatom cells, first of all by their position within the image, defined, for example, by their outline or a rectangular bounding box enclosing them or their center point, and also by one or more labels, which are then giving, for example, the species name of the cell, or describe its condition, if it's a complete frustule, maybe even part of a chain, or only the valve or a fragment, or might refer to the orientation. So if you are looking at the cell from the valve or from the girdle view. And this is really a great technique. I absolutely love it, and I hope I can transfer a little bit of my love for the virtual slides also to you. So here we see a screenshot of the Biigle software, which we are using for annotating our virtual slides. Andrea will tell more about this later. And here in the center, we see such a virtual slide, which already has been annotated. And because of that, it has all of this nice, tiny, colorful spots and tintles. This virtual slide now has a resolution of 65,000 by 25,000 pixels. So it's a little bit bigger than about 1.6 gigapixels, which really is a lot. And these are representing an area of about 6 by a little bit more than 2 millimeters on the physical slide. So actually, it's not even a really large virtual slide. They can become much, much bigger. But for most of the stuff we are doing, something like this size is okay. And this area now has been imaged using an objective with a 63 times magnification and an NA of 1.42. And because of that, we can now zoom into this image and zoom in until we finally arrive at this spot here, where we can now look at a single cell in all its glorious beauty. And also we will see all the fine structural details. So this is really high resolution here. This is about as good as you can get with standard conventional bright field microscopy, plus the benefit of having used focus stacking. But please keep in mind that whatever you see here with all its fine details actually within the virtual slide is just this tiny little green spot here. And this is really brilliant because now suddenly we have something like a digital carbon copy of the original slide, which is showing each and every object contained on the slide. With your traditional approach working on the microscope with the physical slide, if you're taking images at all, you will only have images of some selected cells, and you will not image the area in between. And if you just miss anything when looking at the slide, it will not be included in your analysis. But this virtual slide now contains just everything which is on the physical slide. And so this enables to have nearly any kind of analysis without ever accessing the physical slide again. So the slide can actually stay on the shelf where it's safe, and that shelf might even be on a different continent. For your own analysis, it doesn't matter, because you can do everything digitally and online. But if you want, you can approach this virtual slide pretty much the same way as you are used to with the physical slide on the microscope. So you can change the magnification by just zooming in and out. You can drag it around the screen the same way you would move the slide on the microscope stage. If you do not want to look at all of the virtual slide, but only on a subsection of it, tools like Biigle make things easier for you by offering workflows using, for example, grid or random sampling. Or if you want just the opposite and to make sure that you really saw every tiny bit of the virtual slide, then there is something they call the lawnmower mode, which is just guiding you over the surface of the complete virtual slide, and nothing will be omitted. If you do not do focus stacking, but keep the individual focal planes, then you can even focus up and down through the objects on the slide. So we do not use this technique for our diatom research, but we're using this widely as a digital and online replacement for our 101 microscopy labs in botany, zoology, and microbiology. And so the students can look at the samples and use them pretty much the same way as they would do on the actual microscope, but they can stay at home where they are safe. But let's get back to diatoms and virtual slides. So within the virtual slide, everything is kept within its context because just everything is imaged. And so this enables you to very quickly get a feeling about the sample. And it also very much improves documentation, shareability, and collaboration. Andrea will tell more about this later. Also, it makes it very easy to later have something like a reanalysis. So imagine you're doing a long-term project, and somewhere in the middle of that, you find out that actually it would have been a good idea to differentiate one of the species you investigated into several subspecies or morphotypes or something like that. Or even you would have liked to look at some other stuff on the slide, organisms which maybe were not even diatoms, and so you did not consider them when you started the whole thing. If you did your analysis directly on the microscope on the physical slide, especially if you did not take any images, you maybe would even need to restart the whole thing and just throw away all of the counts you have until now. But if you did everything on the virtual slides, then nothing is lost because you can just keep all of your annotations which are okay the way they are, and just directly target very specifically only the few annotations which need revision, and look at them and change, for example, the taxonomic label or whatever you need to do, and you're done. So this is very, very easy. But virtual slides also are coming with some challenges, and just to cut this very short, usually the image data contained in them is far too large to be processed by most of the common software tools you might be used to. So you either need to use specialized software, like for example Biigle, or you need to program stuff on your own, which is what we are doing most of the time. All the stuff I showed you until here was about automating microscopy, but still having the analysis done manually. But we can also automate those. And the first tool we developed for doing so is called SHERPA. And this is a tool for digital data morphometry. So what SHERPA does is that you feed it with hundreds of thousands of images, in this case, let's say normal sized images up to a few megapixels, so that this does not work directly on the large virtual slide images. But then SHERPA will go through all of these images and look for objects which are similar to a certain shape or a set of shapes you provided with, marks all of these objects and then measures them by about 30 different morphometric features. And so it measures many, many more features than you would usually do when you're doing manual morphometry. And also it measures everything quite accurately, whilst quite often, if you do it manually, you would, for example, just approximate something like the valve size using a formula. There's a paper out telling everything about SHERPA, which already is more than 6 years old, and so I will not go any deeper into this. What we also had a bunch of investigations making use of SHERPA, and some of these looked into something like 10 or even 20,000 cells and measured them. And so this whole stuff is really about upscaling diatomorphometry. So we are measuring much more cells by many more features, and we measure those features even more precise than it was done before. But whenever I advertise SHERPA, people usually approach me and ask for something else they wanted, which is just automating diatom identification. So they wanted to have something like automated abundance counts for upscaling their investigations. And if one wants to have this stuff, it's a good idea to split it up into 2 different tasks, where the first is finding all of the individual diatoms within an image. The technical term for this is segmentation. And then in the second step, name each of the diatoms you found just by its taxonomic name, which is a classification task. And the first step, the segmentation, is a very generic task. It's quite easy to learn, at least for humans, not necessarily for computers. So I think most of you will agree that you can teach a student maybe within an hour or maybe even just within a few minutes to look at a slide and tell apart most of the stuff on that which is diatoms from most of the stuff which actually isn't, maybe with a few exceptions which are a little bit more difficult. But you do not need any deeper taxonomic knowledge to do so. But the taxonomic classification then is a really highly specific task, which is really expensive to learn. So if you remember your days as a rookie in diatom taxonomy, it probably took you something like a few months to become really comfortable with this kind of work. And on top of that, it might even have taken you several years to become really, really good at this. And even then, we're talking about only a small subset of the 10 to 20,000 diatom species which have been described until today, especially considering that there's another 100,000 or even maybe a few million more species expected to be out there somewhere. Automating this kind of stuff has been addressed every now and then for several decades now. I already told you about this EDIAC project. But whatever was attempted in the last few years usually used methods which were based on so-called deep neural networks. So this is this deep learning stuff everybody is so excited about nowadays. Deep learning is a subdiscipline of machine learning. So we are talking about methods, or to be more precise, models, which teach themselves how they should do things, but you need to teach them by examples what they should actually do. The first of our 2 tasks, the segmentation, just as a reminder, is about finding individual diatoms, and we want to do this automatically within the image data. And then for each of these cells, we want to determine their position within the image and also the shape or the outline. To make things a little bit easier, we will only discriminate 2 classes, which are anything which is a diatom versus anything which is not, which is, for example, debris, non-diatom fragments, but also smaller fragments of diatom cells, which we are not interested in, and also the anti-image background. All of this is not really relevant to us. But on the other hand, to make things a little bit more complicated, at least for ourselves, our taxonomic focus has changed very much in the last 2 years, because before that, we were part of the Alfred Wegener Institute for Polar and Marine Research. So we mostly investigated polar marine diatoms. And now we're proud members of the University of Duisburg-Essen, where we are focusing more and more on local and, thanks to Andrea, also Spanish rivers and lakes. So nowadays we are mostly looking into limnic samples, especially epilithic ones. And these contain much more debris, non-datum particles and fragments than we were used to. Here for a comparison, on the left you see a typical Southern Ocean plankton sample, which is very clean. And here on the right, you see one of our limnic samples, which contains a lot of, well, let's just name it dirt. And these images here actually now are much more complicated to analyze. And actually, it's nearly impossible to automatically process them with traditional image processing pipelines as they are implemented, for example, in SHERPA, but also in hundreds of other image processing tools. But in the last few years, methods have popped up which can handle this kind of material from the area of deep learning, for example, UNet or mask-R-CNN. And these are orienting now on the object texture. So they are looking into the images if they can somewhere find specific structural features, like, for example, the valve ornamentation, to tell if at any position there is a diatom present or not. And if they found a diatom, they know from their training data how their shape should be. So they are more or less robust against the dirty image background and can determine the cell's shape, even if it's somehow occluded by a piece of debris or whatever. As a drawback, they are not as precise as the traditional methods when it comes to the segmentation in many cases, but for most of our applications, we do not need to have that precise really down to the single pixel level. So that's okay for us. I will now show you some of our work which currently is in progress. So here you once again see a virtual slide of about 1.6 gigapixels. And this actually is my very first attempt on segmenting a whole virtual slide. So because of that, this is still Southern Ocean plankton. And after segmentation, it looks something like this. And now everything in white is supposedly diatoms, anything in black most probably isn't. And if you look into the details now, we can see that for many of the cells, this works really nicely. Sometimes there are minor issues, and sometimes there are major issues. And also some of the debris or smaller valve particles are segmented, which we actually do not want to have. And just in case you're wondering about the two silicoflagellates here, so those are segmented because we deliberately decided to also include them into our investigation, just because we have so many of them within our Southern Ocean plankton samples. The next step, the classification, is about determining the species of each of the individual datum cells which have been found in the segmentation step before. And I will make a little bit of a fuss about the term classification because usually biologists use it in a very different way than data or computer science people use it. So just to be specific with classification, we are referring here to label. It's another one of the statistical terms, each of our diatoms with the correct taxonomic identification, which is the species name. We are by far not the only ones trying to automate this, and especially the Spanish VisiLab group is very busy on this and has some papers out on this too. These deep learning classification models have a problem, which is that they usually will process one input image as a whole and then produce one single classification result for it. And this will give you problems in a situation like this one, where within one image cutout, you have multiple datum cells, because if now the classifier model will produce one single species name as a result, then you have no idea if this belongs to this cell or to this one or to this one or to whatever is present in the image. But if from the segmentation step before, you know at least roughly the object outlines, then you can use that information to mask out anything but one individual cell. And if you now run this image here through the classification model and get your species name predicted, then you can be pretty sure that it belongs to this cell here. A few months ago, we published a paper where we achieved a better than 97% classification success on planktonic Southern Ocean samples using really standard off-the-shelf convolutional neural networks, like, for example, VGG16. But I have to admit that this paper here is not a real-life application from the perspective of a biologist or taxonomist, but this is more from the point of view of data science. And so what we're doing in this paper is that we are investigating the effect of, for example, different model architectures and also the size and composition of the training data on the classification performance. So this paper is giving some of the baselines if you want to implement something like an automated diatom identification with deep learning. But it also will tell a little bit about the workflow we implemented for producing the deep learning data from our virtual slide images. All of this stuff works pretty nicely, but there still are a lot of challenges to overcome. And the major problem is that for automating this stuff with deep learning, one needs really large amounts of well-curated training data. With training data, I'm meaning stuff like this. So one data point in the training data consists of an image cutout depicting one diatom cell, plus the nicely segmented outline, which you need to train your segmentation models, plus a species label, which you need to train the classification model. And the outline and the species name need to be really high quality, because this actually is the ground truth you're using for training and also for evaluating your deep learning models. And so they should be very, very good. Our recommendation is to have something like 100 examples per species you want to classify. So 100 different of these here, which really is a lot. But this, even though it's a very rough number, is in line with what, for example, the Biigle people are doing. The reason why you need so many examples is that species which are underrepresented in the training data quite often are misclassified. But just for the reason that the model does not know them very well, or maybe does not know them even at all. But usually even the misclassifications are given by the model with a very high confidence or probability. And so it's very difficult to tell apart the misclassifications from the correct ones. One way of alleviating the problem of misclassifications is to have the training data taxonomically balanced. Problem with this is that natural species assemblages usually are very, very imbalanced. So here is, as an example, one of our local logos we are investigating with something like 100 different species, and only very few of the species completely dominate the counts, whilst most of the species are represented by only one or two cells. And if you want to boost up all of these counts to 100, then this would really take a major effort. To make things worse, the training data also should cover the morphological plasticity of each of the species, which can be quite high for diatoms. So here, as an example, you have my most favorite diatom, which is Flagellariopsis kerguelensis. It's Southern Ocean plankton. And here the cells can vary by much more than one order of magnitude. And you need to cover this within your training data. But the cells cannot only vary in size, but also very much in their shape. So here you see from this species two different morphotypes. In the upper row, the cells are a little bit more elliptical, and in the lower row, they are more lanceolate. And actually, we found these two morphotypes in the species using our digital methods and digital morphology, morphometrics. And these morphotypes have been overlooked before, even though the species itself is very well investigated because it actually is one of the major players in the global silica cycle. And also this very material has been investigated very, very closely by many people because it comes from one of the standard sediment cores many people at the Alfred Wegener Institute are working with. Nevertheless, these two morphotypes have been overlooked before. So this is some of the funny stuff you might run into if you're using this digital methods to improve your research. And actually nowadays, from what we know, it seems like these actually are not even two different morphotypes of one species, but they seem to be originating from three different species. But let's get back to our deep learning training data. So to make things even more worse, the training data also needs to cover all of the variability you have in your image quality, depending on the camera or the microscope setup you were using, or the lighting, or whatever. And this now closes the loop back to the beginning of the talk when I introduced you to why we're using slide scanners. One of the reasons is that they are getting us rid of this problem because the image quality is very, very standardized with slide scanning. This actually is most of the stuff I wanted to talk about today, but there's one topic open which is really a matter of the heart to me, which is how we can facilitate this deep learning methods for automatic diatom identification just for everybody. Because actually these methods are very easily available. There's a lot of software libraries out there or even ready-to-use systems where you do not need to program anything on yourself, which can implement at least the standard models very, very easily. So if you want to do it yourself, with something like 20 or 30 lines of code, you can implement your deep learning model, you can train it with your data, and you can use it for classifying some image data it has not seen before. So that is broadly available, and these methods are really, really good. But the one thing which is missing is the training data. This is the major bottleneck when it comes to deep learning, not only for diatoms. So everybody is in dire need of training data. Few people have it, and really nobody has enough of it. Already many, many years ago, our group decided that along with each paper we are publishing, we will also publish all of the data, which nowadays is also the training data we produced. And I would kindly ask you to please do the same with your training data, because I'm pretty sure there are some people in the audience today who already are working on this kind of stuff. I know your training data is very, very valuable, and actually it's the kind of gold everybody is digging for right now in deep learning. But actually the lacking training data is the only thing which is keeping us from having something like a diatom identification system which runs mostly automatic and which is good enough to be used for routine work, everyday work, all of your research. And I think we actually could have from the deep learning side, from the program side, a system like that, maybe even within a few months or maybe a year. But the only thing right now keeping us from having it is the lack of training data. And so maybe we as a Diatom community should make up our mind if it's not a good idea to have something like a central open repository where just everybody can store their training data and just anybody needing it can retrieve it then again. And those now are my final words for today. If you have any questions, we will have this discussion later, but I will pass on the talk now to Andrea.
Okay, so now onto a little bit more from a view of a diatomologist, so to speak. So how does it work for me? The idea is that we have used it both in teaching and also in investigating experience. But how do we do it? You will pretty much know the first step. Of course, you sample some biofilm, and then you make the chemical preparation. From there, you make images, either with your normal microscope and just images, and or you use the slide scanner and make a virtual slide, and then you upload it onto a labeling software, in this case, in our case, Biigle, that I will present to you now. From there, when you have annotated, so labeled all of the diatoms names, you can then download the species list, and from there make all your ecological, but also your statistical studies from there. So now a little bit about Biigle. We use this Biigle as an image graphical labeler and explorer. It was made by these fine gentlemen here from the University of Bielefeld, from the biodata mining group. And it is a very fine tool to work with all kinds of images, both single images, but also our enormous virtual slides. If you have interest in looking a little bit more into it, please visit the biigle.de site, where you can read everything up and also create your own user. So as a diatomologist, why do I really want to have some slides scanned and uploaded as virtual slides? Well, for one, you can easily compare different types of diatoms in different sites much more easily than you would having your own usual light microscope. But also, what about making your usual stuff with diatoms? You want to identify them. It's pretty easy using the morphometry based on this kind of annotation software, since you don't only have a scale bar, but also you are able to make direct measurements on the diatoms, as we will see after this in a very short video that I hope I will be able to show you. Also, we can use well, we can label those diatoms using taxonomical label trees that we can create ourselves, and also non-taxonomical label trees. Here we have an example of the freshwater diatom taxonomical label tree that you will remember the structure as the one that is used in diatoms.org from Diatoms of North America. But also another label tree in which more diatom forms are written down, for instance, for students, the question mark, so that they can annotate the ones that they are not really sure of. Also, if you have a particularly good diatom that you really want to put into your next publication, you can label it as good and find it very, very quickly. Or also some usual positional, but also teratologies, and also everything can be labeled. But the taxonomical label theory doesn't have to be like that, but it also can be relatively phylogenetical, so to speak, in traditional classification. If you are interested in updating your own label tree, you can do so with the taxonomical ones using the help of the World Registry of Marine Species, which maybe will not help you much if you are working with freshwater diatoms, but who knows, maybe you will have the possibility of doing so. The idea is that in Biigle you have different types of observers. For instance, a guest could be made so that a person can just look into your diatoms. But very interestingly, there is the user of the position of editor, who can not only make annotations, but also edit and delete his or her own annotations. This is very, very helpful, because this is a perfect teaching tool, because there is also one person above this. This is the admin or expert, who can also delete if these people in the editor positions, for instance, have recognized some debris as a diatom, and so on. Now I will go into some examples. For instance, in collaborative annotation, these are my wonderful bachelor students, Victoria and Lisa, who have been working a lot through this corona pandemic, even though they were able to make to take their own samples and also make their preparation. This is as far as they got before the pandemic really hit, and they had to make all their identifications online. It was very, very lucky for us, since Lisa was stuck in the USA during that time, so that we were able to make intercontinental teaching possible. This was a very interesting experience for us, I think. After that, in between Corona waves, so to speak, we had a workshop for biology students on diatom biology and ecology 101, so to speak. And then we used a little bit of a mixed model. They were able to look into real light microscopes, but the teaching effort, so to speak, so teaching them about diatom taxonomy was made using digital microscopy. We also subdivided them into groups so that they made a whole inventory as a group, and supervision and teaching was made pretty much online. Not only students can collaborate very internationally, as you see, but experts are also able to do so. Here we have Dr. Hannah Hartung, who is also here at the University of Cologne here in Germany, collaborating at the same time with Professor Dr. Jeffrey Stone in Indiana State. And they are able to annotate different diatoms at the same time, pretty much, while sitting in different continents. No problems there. Returning a little bit to the students and the editors, they, or even if you are an expert, you can also have some times where you don't really know what you're seeing. So you have the possibility of either request annotation assistance from the experts, capture a screenshot, or even if you zoom into a particular diatom that you don't recognize, it's really easy to just copy the link, the URL that is above the slide that you're annotating, copy this into an email and send it to whomever also has a Biigle user, for sure, but that can then look into it and say, okay, this is this diatom here. To try to observe how to use it, we have here, for instance, the inventory here on the right of all the diatoms that have been identified in this particular slide. It is very helpful for teaching in this case, but when you are working more in higher volume of data, a very helpful way to work with is this Largo annotation system. This is a tail-based annotation in which a particular diatom is selected, so to speak, and then it is shown as a combination of different tiles, so that you can see whether all of them have pretty much the same form, shape or form, or if someone just jumps out on you. It's pretty easy to see, even though it is relatively small, so you will have to zoom in to look a little bit more. It is, however, very, very helpful for updating taxonomy. You know how diatoms work, and that the diatom taxonomy keeps changing because of the great work most diatom taxonomies are doing. So you just can update everything, no problem at all. Another idea is that you can harmonize diatom annotations. What do I mean by that? There is a thing called the annotation sessions. There you can either divide it by time or by people. So that, for instance, we have here a new annotation section with 3 users. These 3 users don't see the annotations of the other 2. And they also don't see the annotations that they made before. What does that mean? So they could be all annotating the same diatom without seeing that what the other one has done, so that we can see whether they are harmonized, they do the same thing, or they don't. Or even with the same person, you know how it is that you sometimes identify one diatom and the day after that you identified it as something other. This you can also do with this kind of annotation sessions. So it is a very good collaboration tool, a very good review tool, and perfect intercalibration tool, I think, in my opinion. So now I show you a short demonstration. Here you see how the first look of Biigle is. You go into a particular project, freshwater diatoms, and here you have different sampling efforts, so to speak. In each one of these sampling efforts, you have different slides, virtual slides, with all their annotations. Here you have the label trees, where you have the taxonomical and the non-taxonomical label trees, and the members that you can add and also edit if necessary. If you then go to Largo, you can, for instance, as I told you before, select a particular diatom, in this case, the genus Cimbella, and you see everything. So one jumps out. This is this Cimbella, so we will go and take a look at it. We can zoom into the Cimbella so that we see, and we can make all the typical measurements to know which one it is taxonomically. So transapical length, the width, also, whoops, wait a minute, we can make the, select 10 micrometers in order to count all the striae that are necessary. And in this particular case, we even have the opportunity to see the punctae, so that we can also make a measurement here and count the punctae and make the calculations. From there, we can then, using, of course, the bibliography, select the diatom that we have decided it is, and then we can just rename it depending on what we have selected. We can rebrand it, so to speak, so that our previous name is deleted. The interesting thing is that when you do that, you are thrown into the next sampling, into the next name, so that you can see, for instance, if some of them kind of stand out. Okay, and after you have done everything, you are just able to download your data in the form of tables like this, and then you can do everything you were usually doing with your handmade lists. So it's pretty easy to use, I think. So would you like to collaborate with us? We have something for you, because we could scan your slides for you and also update them, upload them, sorry, to a web-based annotation system of your choice, not necessarily only Biigle. But in return, we would like to get to use your annotation data, so that we have the labels and the diatoms in order to show our deep learning methods as Michael described. So please, if you are interested, please do contact us. We are these many people, and we would be very happy to know from you. And well, we also want to just thank the people from our work before, and just thank you for your attention. So please let us know if you have any questions.
Thank you very much, Michael and Andrea. That was amazing. If people would like to post questions for Michael and Andrea, can you go ahead and enter them in the chat box down at the bottom, and we'll go through those. And we've got a few minutes before the top of the hour, so I think we'll have some chance to have some have some good discussion here. Sylvia asks us, how many girdle view images are needed for training data? And do you consider how frequently a species will be observed in girdle view when you're looking at a slide?
Oh, really, it's hard to answer this. So I should have been a little bit more precise. So when I talked about having something like 100 examples per species, that was 100 examples in the vulvar view and 100 examples in girdle view, if you can get it. The more, the better. From our experiments, we know that sometimes it would also work with having like, something like 20 or 30 images, but that depends very much on your species, on the amount of species you're investigating, on how similar they are, and so on and so on. So there's really no general answer to stuff like this.
Okay, thanks. In sort of the same vein, Marina asks us, can any set of images with associated names serve as training data? And are in coming up with these training data sets, is image quality or number of images going to be real critical for developing a training set for learning?
Yes, it is. Just to be precise, the quality is about the properties of the images. So it does not need to be a high-end or super high-end microscope for generating high-quality images. For example, many of the images the Spanish Lab group is using from their optical appearance are much worse than what we produce with our slide scanners. But still, these methods work, and we are also with our slide scanners still busy with honing the image quality because we are having some problems with that. But it's always about having as many images as possible. That is actually the reason why all of you are so busy, for example, with Facebook, Google, and so on, because they are actually harvesting whatever you are doing to produce their own deep learning data. It's only that they are not working on diatoms, but more on stuff which can be used for commercial purposes or whatever.
Okay. Ingrid asks us, you guys brought up a number of different sets of software, Biigle, SHERPA. Are these software free for users?
Yes.
They are. And
okay.
I think Lawrence also asked if it's downloadable. I'm afraid not, as far as I know. Well,
yes,
it is possible, but
So,
it's a little bit more complicated than that.
So SHERPA, for example, you can just download from our website. When it comes to Biigle, it is open source, but it's not a tool that you just download and start. Actually, it's a whole server application you need to install. So that will be a little bit more complicated, but it's completely for free, and just ask a computer expert if you want to set up this by your own. But also the Biigle people, also they developed Biigle not to use it for their own biological research, but for harvesting deep learning data. So they are data science experts. So usually you can just ask them if it's possible to get access to their instance of Biigle and just upload your data there. And usually they are really happy to provide you with access.
Thank you. Thank you. Sylvia asks us, you guys have talked about huge amount of images, huge amounts of computing power that are necessary. How are you storing so many of these large files for the project? Are they on servers that are supported by university, grants, sources, all of the above?
Yeah, so we're just building up our stuff here at our new university. So at our own institute, we had a file server for our own. Now we're using just the IT resources of the university. Until now, the amount of data we produce can be handled by ourselves, but we will very soon reach a point where we really need the help of some major IT storage infrastructure.
Okay. Yanni brings up an interesting application of this. How could citizen science or the people who are just passionate about diatoms contribute to these types of projects?
They can annotate images if they are, well, let's be honest, good enough with the taxonomy. I know for hobby diatomists, it's quite common that they are actually very good with this kind of stuff, and if they're interested in annotating data, then they should just be given the possibility to do so. Okay. I see not much of a problem there.
Ingrid brings up the slide scanning. She asks, how long does it take to create a slide scan? And I want to follow up with that. With many of us have awesome microscopes sitting in our labs right now. Are slide scanning accessories available for most of these microscopes, or is it all, we got to start over?
Well, you're addressing my hobby project for the next 3 to 5 years, because I actually would like to build something which you could just place on your already existing microscope to convert it into something like a slide scanner. The Spanish Biigle Lab people, they converted a conventional microscope in something like an automatic slide scanner. But here again, there's a problem with the mechanical precision. So they can somehow remote control the microscope, but as far as I know, they are not able to have something like a complete slide scanned, just because of the precision problems. So the actual microscope stage, which is attached to your microscope, you can automate that using just stepper motors, which will turn the knobs and wheels. But the mechanics is not precise enough to, for example, revisit a position. If you just move the knob by something like 360 degrees and move it back by 360 degrees, you will still be off by something like 100 micron or whatever. And that is not precise enough to do slide scanning. With the commercial systems, this differs very much on which system you use. So I showed 2 pictures in the beginning. One of them was the Metasystems Metafair, which we used for something like 10 years until a year ago or whatever. And that actually is built on the base of a conventional Zeiss axioscope, which is only extended by having a automated slide stage. So that can be computer controlled. But everything else is pretty much available off the shelf. Also, these XY stages from, for example, Merzhäuser are available. They are not cheap, but they are available. So this old Metafair system is really brilliant. I absolutely loved it. But a few months ago, we switched over to this brand new Olympus VS200. Actually, as far as I know, we were the second worldwide who bought the system. And this is something like about 10 times faster when it comes to scanning. So we can scan something like 5 by 5 millimeters, which is our standard size we are using, in 60 different focal planes in something like 1 hour.
Okay.
So
wow.
One can debate or discuss if this is really high throughput, but you can do something like 5 to 10 slides within a working day. Also, you can feed, I think, 6 slides into it and then have it run overnight. So this can really work something like 24/7. If you have a little bit more money, you can even attach a automatic slide feeding system where you can store something like, I think, 320 slides, and then all of them are run through automatically. This will be a little bit complicated with the Diatom samples because their software currently focuses on something like tissue samples from pathology. So for the system now, it's in its current version, it's hard to identify where on the slide is the cover slip with the tiny diatoms. That's why we are still starting each of the scans manually.
Okay.
But hopefully they will get there at some point.
How Ewan asks us, how do you deal with something like Applicatio, where you may have many cells still connected in a colony?
Yeah, great question. No answer. So we're just investigating all of the methods, and currently our work is focusing on anything which will just fall apart into individual cells, or maybe into very short change of 2 or 3 cells. But no larger chains, no colonies, whatever. It would be worse to try stuff like this.
Mm-hmm.
We just haven't done it before. So usually these deep learning systems are very powerful. It's a little bit more complicated when stuff is very crowded on the slide. Then you would need to use slightly different methods. But yeah, as I said, we did not investigate stuff like this until now. So I cannot really tell how good it is.
Okay. Dick Gordon brings up another way to think about this, and whether or not this could be adapted to time-lapse analysis. Is that something that has been attempted?
Not by us. So on the slide scanner, we are usually only scanning permanent slides. So time-lapse would be of not much use there because nothing moves. On top of the slide scanner, there's a camera attached, which is pretty much standard, so that can do something like 20 to 30 frames per second. Okay. Just like with any microscope which has a digital camera attached, you could probably do something like time-lapse with use, but I do not think that for our slide scanner, this is supported by the software, because you can have that much cheaper than investing something like $100,000 into such a slide scanner. They are really expensive.
Okay, let's take one last question before we end our presentation. It's going to be the hard question, though. How do you deal with these tiny, transparent little those really hard diatoms like fistulifera that are just hard to see, hard to find anything to focus on, and are tiny?
Once again, really good question. Not an answer yet. So as I said, this is all stuff we are working on. We did not look into hundreds of thousands of species until now. We will address this more in detail with a project which we are just writing a grant proposal for. Maybe Andrea can tell a little bit more about what she sees in her limbic samples. Maybe there's something similar.
Yeah, I have found fistulifera, and I also try to create as many examples for the deep learning networks to learn that. We just don't know how many we will get in order to show it to it and to it being able to identify it. That's the problem. So not easy, but we're trying.
Well, I want to thank all of our participants who joined us today, and especially Michael and Andrea, for a wonderful presentation and helping us think about diatoms and looking at them in such a new and novel way. I appreciate your offering to help out with the Diatom Web Academy, and want to remind everyone that these presentations are recorded, and you can go to diatoms.org at the news page to find links to all of them, or to our YouTube Diatom Web Academy channel. Thanks everyone for joining us today.
Okay,
Thank you.
thanks.
Bye-bye.
Stay well and safe.